Welcome, deep divers, to another journey. We're diving into the incredibly small yet really impactful world of genetic variations.
That's right.
Today we're looking at molecular markers, specifically one type, single nucleotide polymorphisms, SNPs for short.
Yep, SNPs. And you sent us some great material, a presentation, a course transcript.
Exactly. So our mission today, cut through the complexity.
Yeah, we want to unpack how these tiny, tiny DNA differences are found, how they're analyzed, and crucially, how they're being used, especially in things like plant breeding.
We'll look at how sequencing tech has evolved, get into the latest genotyping methods.
And see how it all helps us understand genetic diversity and, improve crops. Should be some real aha moments.
Okay, let's start right at the beginning then. What exactly is SNP? Lay the foundation for us.
Right. So the basic idea is that all molecular markers, they come from mutations, differences in the DNA sequence, variation.
Okay.
And SNPs are, well, about the smallest variation you can get. It's a change in just one single nucleotide, one letter at a specific spot in the DNA.
So if you line up DNA from a few different people or plants,
Exactly.
SNP is just one place where, say, one has an A and another has a T.
Precisely that. At the exact same position in the genome.
Yep.
We also see things called indels insertion deletions.
Oh, yes, indel.
Yeah, where there's maybe a small gap because a few bases are missing or maybe extra ones are inserted, but SNPs are usually the focus, mainly because there are just so many of them.
And why should this matter? Why focus on these tiny single-letter changes?
Because despite being small, they're incredibly powerful. Think of them as really precise genetic signposts.
Signpost, no.
They let us tell individuals apart, track inheritance, and importantly, link genetic variations to actual traits we care about, like disease resistance in a crop.
Got it. So tiny differences, big implications.
Yeah.
But how do we even find them in the first place? You can't just see them.
Right, you need to find them first. And the main way, the primary way, is through DNA sequencing, reading the code.
And the first method for that was Sanger sequencing.
Yes, Sanger sequencing. Developed by Fred Sanger, it was the breakthrough method. It could read maybe 700 to 1,000 base pairs.
Which sounds like a lot, but
in the context of a whole genome, it's not much. And crucially, it was very, very expensive back then and pretty slow for large-scale work.
how did it actually work? What was the clever bit?
It's a chain termination method. So you start by breaking the long DNA into smaller bits.
Okay, manageable pieces.
Then you clone these pieces into plasmids known DNA circles inside bacteria. The bacteria multiply, making many copies of your fragment.
So you amplify it first.
Kind of, yeah. Then you use PCR, but with a special mix. You have the normal DNA building blocks, the DNTPs.
ATCG.
Right. And you also add special ones called DDNTPs, dideoxynucleotides. Each one, ATCG, has a unique fluorescent label, a color. And critically, when a DDNTP gets added to the growing DNA chain,
It stops.
it stops, chain terminated. So you end up with a whole collection of DNA fragments, all different lengths, each ending with a specific colored base.
Okay, I think I see. Different lengths, different colored ends.
Exactly. Then you separate these fragments by size using capillary electrophoresis. Little tubes, electric current. Smaller fragments move faster.
Right.
As they pass a detector, a light source makes the labels fluoresce. The machine reads the color. So the order of colors, C, T, A, G, whatever. That tells you the DNA sequence.
Like runners in a race stopping at different points, and you record their color to know the order.
That's a decent analogy, yeah. Groundbreaking stuff, but like we said, costly and not great for sequencing, say, a whole plant genome quickly.
So that was the bottleneck, cost and scale. What broke that bottleneck?
That would be next-generation sequencing, or NGS. Things like the Illumina platform. This was the revolution.
How so? What changed?
It massively dropped the cost of sequencing, just orders of magnitude cheaper, and hugely increased the amount of data you could get.
But the reads were shorter, right? You mentioned that.
They were, initially. Illumina might give you, say, 80 to 120 base pair reads, much shorter than Sanger. But you got millions, even billions of them. And that short read sequencing is still incredibly powerful for finding SNPs and genotyping.
Okay, shorter reads, but way, way more of them and much cheaper.
Yeah.
How did NGS manage that? What's the process?
Well, you still start by fragmenting the DNA, but no cloning into bacteria this time.
No plasmids.
No. Instead, you ligate or stick known short DNA sequences called adapters onto the ends of your fragments.
Adapters, okay, like little handles.
Kind of. They do two key things. One, they provide a known sequence for PCR primers to bind to later.
Right.
And two, they allow these fragments to attach onto a solid surface, like a glass slide, often called an array or flow cell.
So they're fixed in place.
Exactly. Then, on this array, the fragments are amplified right there, creating millions of little clusters of identical DNA sequences. These are sometimes called polonies.
Polonies, okay. Millions of clusters on a slide.
Then comes the sequencing part, which is cyclical. In each cycle, you flow over labeled nucleotides A, T, C, G, again with fluorescent colors.
Like Sanger.
Similar idea with the fluorescent bases, but different chemistry. Only the correct complementary base will bind to the template strand at each cluster. Then you take a picture.
A picture.
A high-res image of the whole array. It captures the color of the fluorescence at every single cluster, telling you which base was just added. Then you wash away any unbound nucleotides and start the next cycle. Add the next base, take a picture, wash, repeat.
I see. So you're not sequencing one fragment at a time down a capillary.
No, that's the absolute key difference. You are sequencing millions of fragments simultaneously, in parallel, across that entire array.
Parallel processing. That's the game changer for cost and speed.
Absolutely. That's why NGS is so powerful for large-scale genetics, from reading one page to reading a whole library at once.
Okay, so we've discovered these SNPs using sequencing. But then you need to know which versions, which alleles, a specific individual has, right? That's genotyping.
Exactly. Discovery is finding the locations of variation. Genotyping is figuring out what's at that location in each individual you're studying.
And there were methods for this before NGS became the standard for genotyping too. Non-NGS methods.
Oh yes, several clever technologies. One is mass spectrometry.
Mass spec. How does that work for DNA?
It relies on modifying the nucleotides slightly, so they have different molecular masses. You incorporate these into the DNA at the SNP site. The mass spectrometer then measures the mass.
And the mass tells you which nucleotide it is, like weighing them.
In effect, yes. If an individual is homozygous, has two copies of the same allele, say A, you see one signal, one peak, corresponding to the mass of A, but maybe it's stronger.
Double intensity, maybe.
Right. If they're heterozygous, say, they have one A and one T, you'll see two distinct peaks, one for the mass of A, one for T.
Where is this particularly useful?
It's actually very powerful in complex polyploids. The source mentioned sugarcane, for example.
Sugarcane, right, which can have many sets of chromosomes, maybe 10 or 12 sets.
Exactly. So it's not just about A or T. It's about how many As versus how many Ts. Mass spec allows for what's called dosage calling.
Dosage calling. You mean counting the alleles.
Precisely. It can tell you if that sugarcane plant has, say, 9 A alleles and 1 B allele at that SNP location, or 8 and 2, or whatever the combination is. This was a huge advantage over older PCR methods, which often just gave you a yes-no answer, not the proportions.
That's amazing detail for something so complex. What other non-NGS methods were there?
Chip arrays were, and still are, quite common. Microarrays.
Okay, chips. How do they work?
You have a solid surface, like a glass slide, but it's covered in tiny spots or beads. Each bead has short DNA sequences attached, designed to be complementary to one specific SNP allele, say the A version or the T version.
Like little allele traps.
Kind of, yeah. Then you take DNA from your individual, label it fluorescently, and wash it over the chip. The DNA fragments will bind or hybridize to the beads with the matching sequence.
And you look for the fluorescence signal.
Exactly. If the individual is homozygous A, you'll see a signal, maybe red, on the A beads. Homozygous T, maybe blue on the T beads, if they're heterozygous.
You see both, or a mix.
You'd typically see a mixed signal, maybe appearing green or purple, depending on the labels on both types of beads. And importantly, like mass spec, the intensity of the fluorescence can tell you about dosage.
So again, useful for polyploids, like potato.
Perfect example. In a tetraploid potato, 4 sets of chromosomes, the chip array can distinguish between AAA, 4 A's, AAA, 3 A's, 1 B, AAB, 2 and 2, and so on, based on the relative intensity of the signals.
So these methods, mass spec arrays, they could handle heterozygotes and even dosage.
Yes, because SNPs are codominant markers. That's a key point.
Codominant meaning?
Meaning both alleles are expressed or detectable. Unlike some older marker types where one allele might mask the other, with SNPs you can clearly distinguish the homozygous state, like AA or TT, from the heterozygous state, AT.
Makes sense. And there were others too. KASP, TOCMAN.
Yep, KASP, competitive allele-specific PCR, RAMP, TACMAN. Several PCR-based methods, often relying on different fluorescent dyes to call genotypes. You'd see clusters of dots on a graph, maybe red for one homozygote, blue for the other, green for the heterozygotes. Lots of options depending on the scale and cost needed.
Okay, so a whole toolkit even before NGS took over genotyping too. But let's get to the big one now. Genotyping by sequencing, GBS.
Right, GBS. This really became the dominant method for large-scale studies. Developed at Cornell, hugely impactful.
Why GBS? What makes it so important for, someone learning about this?
Because it's usually the most cost-effective NGS-based approach, and crucially, it does SNP discovery and genotyping at the same time, in the same experiment.
Discovery and genotyping together. Efficient.
Very. It essentially made it feasible to analyze the genetics of hundreds, even thousands of individuals across many, many SNP markers, much more affordably than before. It really accelerated breeding programs worldwide.
So how does GBS pull off this simultaneous discovery and genotyping so efficiently? What's the trick?
Two main principles. First, it uses restriction enzymes. These are like molecular scissors that cut DNA at specific recognition sequences.
Okay, chop up the genome.
Right, but only at specific sites. This creates what's called a reduced representation library. You're not trying to sequence the entire genome, which is often huge and repetitive. You're just sequencing the fragments next to these cut sites.
So you're sampling the genome, focusing the sequencing effort.
Exactly. You might only sequence, say, 1% of the genome, but those sequenced bits are spread out across all the chromosomes, giving you good coverage of the genetic variation without the cost of sequencing everything.
Smart. Okay, that's reduced representation. What's the second principle? How do you handle hundreds of samples?
This is where it gets really neat. DNA barcodes.
Barcodes again, like in NGS generally.
Yes, but applied specifically for multiplexing samples in GBS. For each individual sample you want to analyze, you ligate on not just the standard adapters needed for sequencing, but also a unique short DNA sequence, a barcode specific to that individual.
So every fragment from sample 1 gets barcode 1, every fragment from sample 2 gets barcode 2, and so on.
Precisely. Then you pool all these barcoded fragments from hundreds of different individuals together into a single tube.
One tube. Seriously.
Yep. Then you amplify them all together using PCR and sequence the whole pool on an NGS machine like an Illumina sequencer.
Wow. So you get this massive jumble of reads from hundreds of samples. How do you sort it out?
That's where the bioinformatics comes in. After sequencing, software reads the barcode sequence on each read. That tells it which individual that read originally came from. This sorting process is called demultiplexing.
Demultiplexing, okay.
Once sorted, the reads for each individual are typically aligned to a reference genome, if one exists. By comparing the reads to the reference, the software identifies the SNPs and determines the genotype for that individual at each SNP location.
Those barcodes are the magic key to doing hundreds of samples at once. That's huge for cost savings.
It really is. That multiplexing is what makes GBS so incredibly efficient and cost-effective for large populations.
Now, you mentioned aligning to a reference genome. What if you don't have one for your organism?
Good question. There are different bioinformatics pipelines. If you have a reference genome, you use a reference-based pipeline, like Tassel GBS is a popular one. Reads get mapped, variations called relative to the reference.
And if not?
If there's no reference genome, you use a reference-free pipeline, like Unique. This pipeline identifies SNPs by finding sequence tags present in the population and comparing them to each other to find variations without needing an external map. So GBS is flexible.
Let's make this concrete. How was GBS used in, say, a diploid species? The example was wild sweet potato.
Right. Ipomorea trifida, a wild relative of sweet potato, diploid, but highly heterozygous, meaning lots of variation within individuals.
Okay.
Researchers used GBS on a population derived from a cross. They used 96 barcodes per sequencing plate, sequencing nearly 200 individuals together. They chose a specific restriction enzyme, ran it on the sequencer, millions of reads, around 800 million total. These were processed, identifying about 1.7 million unique sequence tags. They aligned these to the Ipomorea trifida reference genome.
Did they all align?
Not quite. About 75% aligned, which is pretty typical. Reasons for non-alignment could be structural differences between the individuals and the reference, or maybe some quality issues in the reference itself.
Okay, 75% alignment. Then SNP calling.
Yep. Initially, they found around 39,000 potential SNPs, but then comes the crucial filtering step.
Cleaning the data.
Exactly. You have to filter out SNPs with too much missing data across samples, or SNPs with very low read depth, which makes the genotype call unreliable. After filtering, they were left with about 18,000 SNPs.
Still a lot.
Still a lot, and then they might filter even further based on the statistical probability of the genotype call, maybe keeping only those with 90% confidence or higher. This resulted in about 1,900 really high confidence SNPs for their analysis.
So getting the raw data is one thing, but the rigorous filtering is essential for reliable results.
Absolutely critical. And importantly, for an outcrossing heterozygous species like this, they used pipelines like tassel-4 poly that perform quantitative genotype calling.
Quantitative, meaning it properly identifies the heterozygotes,
Oh.
A versus AB versus BB.
Exactly. It distinguishes the 3 diploid genotype classes accurately, which is fundamental for things like genetic mapping or diversity analysis in these types of organisms.
That makes sense. Now, what about the really tricky ones? Autopolyploids, like cultivated sweet potato.
Right, the hexaploid sweet potato, Ipomoea batatas, 6 sets of chromosomes, much more complex than diploid.
6 sets. How does GBS handle that?
It's challenging, but doable. Again, GBS was applied to a sweet potato population. They actually used the Ipomoea trifida genome as a pseudo-reference because a high-quality hexaploid reference wasn't available there.
Using a relative's genome.
Yep.
Clever. Did they need more data?
Yes, typically for polyploids, you need higher sequencing depth to confidently call the dosages, so they used more sequencing lanes, getting over 1 billion reads.
1 billion reads.
Yeah, resulting in about 3 million unique tags. The filtering was also stricter. They required at least 50 reads per SNP to make a confident dosage call.
50 reads minimum.
Okay.
And result.
After all the filtering and quantitative dosage calling, they obtained about 4,200 reliable markers. And the key here, again, is the dosage information.
Right, not just AA or BB, but how many of each?
Exactly. For a hexaploid, GBS pipelines designed for polyploids can distinguish between Traco-As, 6As, Bs, APAB, 5As, 1B, AAABB, 4As, 2Bs, and so on for each SNP.
That's incredible resolution. That directly tackles the limitation you mentioned earlier with older methods.
It does, and this dosage information is absolutely critical for genetic analysis in polyploids. We see similar GBS applications succeeding in tetraploid alfalfa and even complex sugarcane, which is roughly, 12-ploid. It shows how the technology and the analysis tools have been adapted.
It's a fantastic example of pushing the tech to handle tough biological realities,
Yeah.
providing unprecedented genetic detail. Now, GBS is dominant, but it's not the only NGS-based method, is it? Are there variations?
Oh, yeah, several variations on the theme exist. There is RAD-seq restriction site, associated DNA sequencing.
RAD-seq, similar idea.
Similar principle, yes. Using restriction enzymes for a reduced representation library. Sometimes it uses 2 different enzymes, DD-ADSEQ, double digest RAD-seq, and often uses paired end sequencing, where you sequence both ends of the DNA fragments.
Giving you a bit more information per fragment.
Potentially, yeah. It's been used effectively in things like octoploid strawberry, another complex polyploid.
Okay. Any others? What about targeting specific genes?
Right. If you don't want genome-wide markers but want to focus on specific regions, maybe genes known to be important, you can use capture-based methods, like SureSelect GBS.
SureSelect, how does that work?
It uses baits, short RNA or DNA sequences designed to match the regions you're interested in. You mix these baits with your DNA library, and they hybridize or bind to your target sequences.
Like fishing out the bits you want.
Exactly like fishing. Then you wash away the non-target DNA and sequence only the captured fragments. This enriches your sequencing for the regions you care about.
It makes sense if you have specific targets.
Yeah. The example was tetraploid potato, where SureSelect GBS focused on gene regions and showed really high agreement, 98%, with KASP genotyping results for dosage. It acted as a validation.
A sort of gold standard check.
Right. And there are commercial platforms too, like CaptureSeq, which uses custom probes, often targeting express gene sequences, cDNA, and emphasizes automation for high throughput.
So GBS, RAD-seq, capture methods, lots of innovation, allowing researchers to pick the right tool for their specific question, their budget, and their organism.
Precisely. The toolkit is constantly expanding and improving.
So as we sort of wrap up this deep dive, where's this heading? What's the next layer beyond just single SNPs?
That's a great question. With the power of NGS, especially reading sequences of 80, 100, 120 bases, we're moving beyond single SNPs to looking at combinations. We can identify multiple nucleotide polymorphisms, or MNPs, within a single read.
Multiple changes close together.
Exactly. And even more powerfully, we can identify haplotypes.
Haplotypes. What defines a haplotype?
A haplotype is basically a set of SNPs that are located very close together on the same chromosome, often within the length of a single NGS read. Because they're so close, They tend to be inherited together as a block.
Ah, linked SNPs inherited as a unit.
Right. So think about it. A single SNP is usually biallelic. It has two forms, like A or T. But if you have, say, three SNPs right next to each other, the combination of alleles across those three spots, like AGC versus ATC versus TGT, defines different haplotypes.
So instead of just two options at one spot, you suddenly have multiple possible combinations across that short region.
Exactly. That block of SNPs, the haplotype, acts like a multi-allelic marker. It has more than just two forms.
And more alleles mean.
More information. Multi-allelic markers, like haplotypes derived from linked SNPs, provide significantly more genetic information per marker than a single biallelic SNP. It gives you much higher resolution for distinguishing individuals or for linking genetic regions to traits. It's almost like getting back some of the information richness we used to get from older marker types like microsatellites, but now derived from sequence data.
So haplotypes give a much more nuanced, detailed picture of the genetic variation.
Precisely. It's critical for fine mapping genes, understanding population structure, and really pushing the boundaries of crop improvement and genetic analysis.
Wow. What an incredible journey today. We've gone from the basic idea of a single letter change in DNA
These tiny SNPs.
all the way through Sanger sequencing, the NGS revolution with Illumina, methods like mass spec and arrays, and then the real powerhouse, GBS, for discovering and genotyping these variations.
And seeing how it's adapted for everything from relatively simple diploids to really complex polyploids like sweet potato and sugarcane, getting that crucial dosage information.
It really highlights how these tools are unlocking genetic secrets.
Absolutely. And as we finish up, here's something to think about. We've seen how understanding these tiny shifts, these single letters, can revolutionize something like plant breeding, boosting yield, fighting disease, improving nutrition.
Yeah, the impact is huge.
So if that's possible just by decoding these subtle differences in plants, what does it imply about the sheer depth of information still hidden within the genomes of all living things, including, ourselves?
How might digging even deeper into these minute variations change medicine, help conserve endangered species, or just fundamentally shift our understanding of life's diversity?
It feels like the deep dive into the genome. It's really only just getting started, doesn't it? Uncovering secrets one letter at a time.